Our study focuses on low-risk MDS, which are patients that commonly show anemia. There is a specific type of patients with MDS that are the MDS with ring sideroblasts, and 80% of them show mutations in SF3B1. SF3B1 is a splicing factor gene that, when it's mutated, will cause the missplicing of genes that are involved in the heme biosynthesis and also in the iron transporter channels. So when this happens, all the heme groups get trapped inside the mitochondria, and this will create this characteristic ring sideroblast in these patients. Then they have anemia. There is like a treatment that has been recently approved from the FDA, that is the luspatercept, and that helps to improve the anemia in these patients in 40% of them. So even though the results are good, there's a need of new therapies for these patients. So in our previous paper, what we found is that when this is happening, when SF3B1 is mutated in these patients, there is an activation of a pathway that it's called HRI or EIF2AK1. When this is up, the cells cannot terminally differentiate, so then we have anemia. So what we found in our paper is that when we depleted this gene with CRISPR-Cas9, we were able to overcome this anemia. So then we started a drug discovery program to target HRI in these patients. And right now we have like a couple of drugs that are like really specific for this pathway. And here is where MDS-RS and sickle cell disease meet, because EIF2AK1 is a key regulator of BCL11A, which is an inhibitor of the fetal hemoglobin. So what we have seen is that when we treat in vitro cells from patients with sickle cell disease, so what we do is we isolate the CD34 positive cells and we let them differentiate for 15 days. And then we treat these cells. So then we are differentiating the cells towards the erythroid lineage. So when we treat the cells with our compounds, we see a nice target engagement, meaning that our drug is targeting this pathway, and we see a downregulation of EIF2S1, that is the downstream effector of EIF2AK1, and then we see a decreased expression of BCL11A and an increased expression of the fetal hemoglobin, meaning that when we are treating samples from patients with sickle cell disease, we are able to increase this fetal hemoglobin with the hopes that this will help eventually with the anemia in these patients. So for us, we are a little biased, obviously, because we are working towards increasing the fetal hemoglobin. So for us, the therapy that it's like probably more exciting are all these new clinical trials that are trying to target either the inhibitors of the fetal hemoglobin, which will be, for example, BCL11A, or those ones that are trying to enhance the fetal hemoglobin. So these are the two ones that we like the most, because obviously we are working on the same, and we are excited to see where all these studies are gonna lead to. So we like these ones right now.